Today we've got the great pleasure of welcoming Keith Fox as our speaker. Keith is Professor of Biochemistry at the University of Southampton in the Centre for Biological Sciences, but he's also an Associate Director of the Faraday Institute for Science and Religion in Cambridge. His career in biological sciences has spanned over 30 years, and he is Senior Executive Editor for the journal Nucleic Acids Research. His research has focused on aspects of DNA recognition by small molecules, oligonucleotides and proteins. But he's always had an interest in the impact of science on society and has appeared on a number of debates on Premier Radio and other radio stations discussing all aspects of science and culture and religion. In his spare time, he is a middle distance runner. It says long distance runner here, but he tells me he doesn't do marathons. No. Ladies and gentlemen, please welcome Keith Fox.
Well, good afternoon. Thank you for that kind introduction. I feel I should add to the beginning, firstly, as a word of warning for those of you who get scared by the word religion in the title. The Faraday Institute only deals with mainstream science and mainstream religion, so there's nothing cranky. I promise you I'm safe. And if it starts to get too technical, I remind you that I am just a pure biochemist. That's all, a simple boy who does biochemistry and tries to think about the way the world works and life in particular. So in addressing this whole topic of genomics, the molecule that comes to mind all the time is this one. And it's the one I've had the privilege of spending 30 years and more of research on. You don't have to know anything about science at all. The man and woman on the Clapham omnibus recognizes this thing. It's DNA, it's a beautiful structure, and it's almost an icon of the age. The only molecular structure in biochemistry that everybody knows, not all the proteins that we do as well. And it's become used in a whole variety of different ways. They've got nothing to do with science at all. So you can find it on postage stamps. It looks quite nice and things to celebrate from it. You can buy DNA perfume for him or for her. And the observant amongst you will notice that actually comes in a three-stranded helix in the bottle. Somehow it's used as a marketing tool. I have no idea how. Well, here we are, the new marketing DNA. So it's something that everybody's familiar with. Its history goes back to the late-ish 19th century. If I ask most students who discovered DNA, they all say Watson and Crick, and no, they didn't. Friedrich Miescher discovered it. A good example for a student and the supervisor. His supervisor refused to publish his work until he'd actually repeated the experiments because he didn't believe what he discovered in terms of this polymer. 1869 come through to the early 20th to middle 20th century when the importance of DNA was recognized as the genetic material. For a long time it's thought that proteins were the things that encoded all the important molecules of life and that DNA was just sort of hanging in there. Through to the now infamous 1953 Nature paper, Watson and Crick elucidating the structure of DNA. And one of them is reputed to have said at the time, we have discovered the secret of life. I think that's rather an overstatement. I think they've discovered the secret of molecular recognition and of how DNA replicates. I hope there's more to the secret of your life than simply a bit of biochemistry, even though it is really fascinating, I do assure you. Through to, this is after I did my PhD, the ability to sequence DNA. And this is Fred Sanger, brilliant biochemist, won 2 Nobel Prizes, one for sequencing protein, another one for sequencing DNA. And this was in 1977, ability to sequence a small bacteriophage, so a phage, a virus that infects a bacterium. And from then on, of course, all the sequencing technologies developed right through to, in 2000, the first draft of the sequence of the human genome was released. All 3 billion of those base pairs of A, G, C and T, and that special sequence, and our knowledge from then on of the things that make up the human genome. It's interesting that when that sequence was released, with press conferences between Washington and London, with all the eminent players involved in that, the sorts of language that was used. So here is Bill Clinton. At the time today, we're learning the language in which God created life. We're gaining ever more awe and for the complexity, the beauty, the wonder of God's most divine and sacred gift. And Francis Collins, who was director of the Human Genome Project at the time and now director of the NIH, talks about how humbling and awe-inspiring it is to realize we caught the first glimpse of our own instruction book. We as humans are the only organism on the planet that's sequenced its own genome. And along with that came some worries. And there were some promises that were made at the time. The president, and that's the president of the US, Clinton, promises that this genetic information must never be used to stigmatize or discriminate against any individual or group. And we'll come back as we go through this talk as to whether that promise has or will be kept through time, and whether the potential is that we could end up stigmatizing people. How many times have you heard the phrase or read the phrase, "it's in their DNA"? The media easily grab hold of something and say it's the fault of their people's DNA. So you can look at the list there. This is something came from my colleague in Cambridge, Dennis Alexander, who collected these over the years. The love cheat gene, the gangster gene, the mean gene. From genes to hormones, biology may help to shape political behavior. And I'll look a little bit at that in a moment. Cigarettes, it's all in your genes. The gluttony gene, the liberal gene, the happiness gene. And one wag wrote to The Guardian in a letter and suggested there was something called the geneticism gene, a gene that predisposes some people to think that behavior is determined by genes. And those who like getting money out of alumni pretend that actually being generous is in your genes. This is a Canadian university, sorry, it's Boston State University. It's in their DNA, building relationships. They build on great relationships. The liberal family tree, it's in your DNA, isn't it, to be generous. If you gather nothing else from this afternoon's talk, please can I encourage you never to use that phrase. It's in somebody's DNA. Corporations use it, newspapers use it, and it rather suggests that we're a kind of determinist organism, that we've got no free will, no choice in what we do. It's a kind of inevitability. So please encourage others, don't use that phrase, it's in their DNA, but I guarantee I have no impact in that at all. With that, we also want to know what makes me, me. My genome could be sequenced, yours could be sequenced. It'll be 99.9% similar to the person next to you, be 98% similar to that of a chimpanzee. What is it that makes me, me? Here are 6 male generations of foxes, my family tree. It's me up there, my dad, my grandfather, and so on. What that's me can I blame on these my antecedents, my ancestors? There are some things that are clearly not genetic. If you look, there are bow ties, neckties, and no ties. That's cultural. All of those, one way or another, were involved in engineering science as their academic, or these weren't academics, but they practiced effectively as builders and engineers. Is that in their genes? Location, they all came from the same part of the country. At least 5 of them did. My son was born in Cambridge. I was born in Bristol, as were all the others, hence the twang that you might hear occasionally. And you can send your DNA off to a company, a reputable one, 23andMe. You may see adverts for them on the train sometimes, and they will tell you lots of things about what's hidden in your genome. One of my colleagues recently sent off for one of these, not through this company, Life Profile. I'll be careful what I say. That the it tests you as to whether you've got BAM, strength, Eureka, intelligence, or Zoom, speed. The superhero assessment decodes secret information in your unique DNA, giving you unprecedented insights into where your superpowers lie. I have no idea what genes they're testing. My colleague has actually sent off a saliva sample, and we'll see what comes back as to where he is on the spectrum. They're very careful, though, in the very small print right down in the bottom of this, that even I can't read here, to say this product is not a diagnostic test and cannot predict your future health. Use of this product is not intended to be a substitute for professional medical judgment. Please consult your doctor if you have a question about a medical issue. And we'll come back to this as we go through as to how much of the things we know about our genome should then be relayed to us through genetic counsellors rather than just something that comes in the post of a box off a swab. James Watson, co-discoverer of the structure of DNA, is reputed to have said, "We used to think that our fate was in the stars. Now, in large measure, our fate is in our genes." Again, I think an overstatement, a rather deterministic thing, that we are nothing but a bag of genes. But there clearly are some things that our genomics, our genetics says that is in our genes. Genetic disorders or diseases, whether they're of metabolism or structure, if you've got one of those, and that's only a very short list of the many thousands of genetic disorders that have now been characterized, then of course you will suffer from some ill health or some disability. And for a variety of different reasons, because the DNA that we have suffers mutations, which are carried from generation to generation, as well as those are point mutations, things like cystic fibrosis, sickle cell anemia, thalassemia, as where one base pair or just a few go wrong within the genome and lead to a disease. Whether there's other ones where the DNA rearranges on the chromosomes, a bit breaks off from one chromosome and attaches to another one in the wrong place, and a number of cancers particularly arise from that. Or whether it's just expansions in the genome, repeat sequences that somehow stutter and get repeated hundreds to thousands of times. And there's a few examples of that: Friedreich's ataxia, Huntington's disease, or a particular form of motor neurone disease. And in the UK, we test newborn babies for the presence of some metabolic diseases, 6 metabolic diseases and 3 other ones, and those are their incidence. And we test them at birth, but they're only the tip of the iceberg of the many thousands of potential but rare genetic diseases that occur. The second genome ever to be sequenced was that of James Watson himself. Now it's much more routine to sequence them. These things originally cost millions of pounds. Michael Egholm was one of those involved in the sequencing of that. At the end of it said, quoting in Nature, "It was so profound how little we were actually able to say to him. To me, it really proved that this is the beginning and not the end. We've got all this data. Now what are we going to do with it, and what does it say to the person whose DNA we've sequenced?" And that was 10 or more years ago, or 8 years ago, sorry, and now we know a lot more about what our sequences involve. Let me look at just a few examples. Here's the sickle cell mutation, a single point mutation in the DNA. A T goes to an A. That leads to a change in the amino acid in the protein, that leads to the change in the structure of the hemoglobin, and the normal cells, red cells, become sickled, and that's sickle cell anemia. Wouldn't it be nice to have a mechanism of just slipping that out and replacing it with the wild type, the normal form? What about something like HIV? We know how that infects cells. It attaches to receptors on the surface. One of those receptors that's essential, we know you can mutate out. There are some people who are resistant to HIV because they have a mutant or missing form of that receptor. They're physiologically quite normal. Wouldn't it be nice that we could just mutate out of that receptor and then it couldn't be infected by HIV? And until very recently, the answer to that was no, you really can't do those sorts of things. Those gene corrections are they're very, very difficult to do, incredibly expensive, and can never be properly applied. But in the past 5 years or so, in particular since 2012, 2013, there are a number of technologies that have been developed for being able to snip into any genome, really, at will, wherever you want, and either inactivate or replace a defective gene. TALENs were the first to be discovered, sorry, ZFNs, zinc finger nucleases, the first. Then came these things called TALENs, and the one that now is widely used and the easiest to use of them all is this thing called CRISPR-Cas9. I know it sounds like a cooking oil, but it is a precise molecular technology. Developed in 2012, 2013. Now it's a fairly simple thing, can be applied almost in any molecular biology laboratory. In fact, there's papers out that suggest that some people could do it in their garage biochemistry, over the kitchen sink almost. It's cheap to do, it's easy to understand, and it can be widely used in a whole lot of different technologies. I checked this morning and I found 4,500 citations to the words CRISPR in either the title or the abstract of papers published since this was discovered less than 4 years ago. So it's a massive technology. And I won't go into the technology, but essentially you design a piece of RNA, a close cousin of DNA, that matches the sequence that you want to tinker with, and inject that into cells, and the enzyme then goes away and does its business. It cuts the DNA, excuse me, the genome wherever the enzyme is saying it should go to, cuts it, and then one of two things can happen when you cut. The cells automatically repair that cut, but they all do it with inefficiency, so miss a few bases out, and you get an indel, a bit's missing, and inactivates the gene. And that has been done for inactivating the receptor for HIV in cell lines. Or you give it the piece of DNA that you would rather have there in its place, and the cell has mechanisms for homologous recombination for just slipping that into the cut, and you insert a piece of DNA wherever you want it. In theory, a very, very simple technique to use. And I say it's been done for establishing HIV resistance within cell lines, and I'll show you a few other examples as we go through this afternoon. Here's a rather moving case reported in New Scientist and in their daily news of a child who was in the terminal stages of leukemia. And she was treated with a T-cell line that had been modified in vitro so as to inactivate certain genes and activate others, and was she was infused with that modified cell line that was coming off the shelf in the molecular biology labs, and it was in remission. I don't know, I can assume that she still is in remission. But a remarkable medical advance from the use of this technology to alter cells and to alter them in vitro and then put them back in the patient. But that, of course, is modifying a somatic cell, one of the cells in a developed organism. Somatic cells, this is what we might call body cells, only affect the individual in which you make that change. It cannot be passed to offspring. The technology, though, could be applied to germ cells or to early embryos, affecting the gametes, the sperm and the egg. And then that will affect not just the individual person, if they're implanted and develop, but could then be passed on to future generations. This is an irreversible change that would affect generations afterwards. And the genie is, if you like, out of the bottle. In 2015, a Chinese group published a paper where they had done just exactly that and reversed the thalassemia mutation within human embryos. It did it with fairly low efficiency, and they did it with embryos that could never be implanted anyway. They were pronuclear ones. In other words, they were cells that had been, they're eggs that had been fertilized with 2 sperm and therefore could never develop. So there was never any intention of implanting these, but at least they proved in vitro that it's possible to edit the human genome. And here are the words of an Oxford biochemist writing. He's written a book called Redesigning Life, John Parrington. The words revolutionary and breakthrough can be overused in media reports about new scientific discoveries. But every once in a while, a scientific discovery is made whose impact on society is likely to be so immense that even an abundance of superlatives may not do it full justice. Genome editing looks set to be such a discovery. And we cannot put the genie back in the bottle. We may have legislation for how it can be used, but this is big stuff, both in the molecular biology laboratory, where you can modify bacteria, plants, cell lines, human cell lines. So let me just take a little bit of a sidestep for a moment, the sorts of things that we might want to do as a result of what we know about genomics. By sequencing genomes of people and other organisms, we can compare their DNA sequence. How does your sequence compare to the person next to you? How does it compare to other primates? How does it compare to a banana and a jellyfish and an E. coli? And we can learn things about that. And you can use that data in a number of different ways. You know, forensics, we're familiar with the sort of screening, the DNA fingerprinting in terms of scene of crime or paternity testing. You can use it for determining susceptibility to disease. Are you the sort of person who's going to suffer from a heart attack? How should you adjust your diet and on the knowledge that you have about the sorts of enzymes that you've got that can metabolize different things? It asks questions I've already said about genetic counselling. If you know about your DNA sequence, if you know what diseases that you might be prone to, you need somebody to be able to give you counsel as to what you should do with your life, and maybe who your partner should be, because what diseases are your offspring going to have? It raises questions about who has access to the data, and I'll come back to that in a moment, and it raises issues about prenatal screening of embryos. It is Christmas, so I've put in a few cartoons that are Christmas related. Dear Santa, thank you for the chemistry set. It appears that you have exactly the same DNA as my father. Easy to genotype on the basis of a small DNA sample. The genetic knowledge that we have from all this, it brings with it a tremendous burden of who should know that. One assumes that the individual themselves has asked for it, so they clearly should know. But would they want to know? I was in a conference where one of the first tests for the ApoE gene, which is one of the predictors of Alzheimer's, was released. And a conference of 300 geneticists said, if any one of you wants to have that test, it's yours for free this afternoon. And nobody took it. And you can see why. There's no treatment. Would you want to know and live in fear for the rest of your days? Should your family, your parents, your children know? If you have a genetic condition hidden in your genome, should you tell your kids? Because it's going to affect them, or it may do. And they would live with that cloud over them. Should you tell your GP? Should you tell your insurers, who maybe render you uninsurable? Should you tell your employer, are you worth investing in all that training that's going to go for the next 20, 30 years if you're going to drop dead of a disease in 10 years' time? All those sorts of questions immediately come out of knowledge about the human genome. But there are other things as well that it raises that are more sociologically related. The question of, is biology destiny? And notice where that was published. This is an article in the International Socialist Review, not well renowned for publishing academic texts on molecular biology and biochemistry. Trends at genetics, the genetics of politics, discovery, challenges, and progress. Genes for, we'll come back to that phrase again, friendships, association between a certain neurotransmitter, dopamine, and a variant of the receptor for that, and political ideology, something published in the Journal of Politics. So these things are seeping into other realms of academic written life. And an article that was published in Nature reviewing this anatomy of politics says that some of the hype that can go into this, writing here in 2012, the author, and I think exaggerating, says an increasing number of studies suggest that biology can exert a significant influence on political beliefs and behaviours. Genes could exert a pull on attitudes concerning topics such as abortion, immigration, the death penalty, and pacifism. Now, I think this, to say exert a significant influence is rather naughty. To exert a minor influence or have some effect on might be allowable. But it goes on, it's difficult to change someone's mind about political issues because their reactions are rooted deep in their physiology. So give up debating with people, give up trying to change their minds because it's in their DNA, they're programmed. Nonsense, sorry. And think about if you accept that sort of determinism, what that means to the way in which you respond to social issues. If you think that your behavior, good or bad, comes out of your genetic complement, if it's fixed by genes, then you can't change society by changing the social structures. It's the fault of the genetic composition of people. The problems lie not in the structure of society, but in the individuals who make it up. The solution, therefore, is to change or eliminate those individuals, not to change existing social structures. So beware of exactly what the implications are sometimes of if you take a pure deterministic genetic approach. And it relates back, I think, to some of the things that were written in the early 20th, late 19th century. Here's Charles Davenport, one of the leaders in the eugenics movement. Characteristics such as pauperism, criminality, feeble-mindedness were biologically inherited. Letting these people loose upon society and permitting them to perpetuate in their offspring these animal traits. And you may choose to forget, and some of you may not realize that until the mid-1970s, there was US legislation for the sterilization of the feeble-minded, preventing people with poor backgrounds, as they thought. It is better for the world if instead of waiting to execute degenerate offspring for a crime or letting them starve for their imbecility, society can prevent those who are manifestly unfit from continuing their kind. 3 generations of imbeciles is enough, they say. Now, I find that very scary. Now, that has been repealed, but it wasn't until the mid-1970s. As we look into our genomes, we find other things as well, which sometimes, well, we don't like them. Something like 1 to 4% of our genome derives from Neanderthals, those other hominids that we don't really know why they went extinct. And there's a greater amount of Neanderthal DNA in Asians and Europeans than there is in Africans. I mean, the out of Africa hypothesis for human migration, those who came out of Africa, of course, encountered the Neanderthals and one way or another may have interbred with them. As Svante Palber, one of those responsible for sequencing the Neanderthal genome that we now know, is Neanderthals are not totally extinct, he says. They live on in some of us. Now think what the reaction is to that is. Your reaction might well be, oh gosh, I'm not that stupid and thick, am I? But actually, some people turn it the other way up, misusing genetic information. Social conservatives, so these are white supremacists within the US. After the discovery of Neanderthal genome, a majority of bloggers on this white supremacist website congratulated themselves for their genetic uniqueness and claimed that Neanderthal DNA was responsible for their intellectual supremacy and the physical prowess of Europeans. You can make of it what you want, can't you? I mean, it's misusing genetic data. Are there genes for, as we look in the genome, genes for criminality? Ladies, can you stick your fingers in your ears just for a few moments? Because the greatest correlation of anything in terms of behavior and genetics is the presence of the Y chromosome, which is male. Over 90% of the population of jails across the globe are men and not women. They have the Y chromosome. Does that mean that possession of a Y chromosome makes you a criminal? Gentlemen, I hope that's not the case. There may be more men in jail, but it doesn't predetermine you to being a criminal. In fact, there are some people who have 2 Y chromosomes, X, Y, Y. In fact, probably 1 in about 1,000 men have an extra copy of that Y chromosome. And there's no evidence that they are more prone to criminal behavior than those who have a standard XY. If you look back at some of the old literature, it suggests there is a correlation. That has since been discredited. There is no evidence that just possession of a Y chromosome makes you a criminal. But there are some genes that do seem to correlate with aggressive behavior. The gene for monoamine oxidase, that's the enzyme that degrades noradrenaline, that fight or flight hormone, has been known as the warrior gene. The more noradrenaline you've got, the more hyped up you are, the more likely you are to be aggressive and vicious. And there's a case study of a Dutch family for which there was a long history of male aggressive behavior, also mental retardation went with that as well, in which there was a point mutation in the gene that codes for that enzyme, so that the enzyme was not made, and their total absence of that particular kind of monoamine oxidase. And the net result is that they had violent behavior across very number of generations. It's on the X chromosome, so it's only seen in males. It is extremely rare. In fact, I'm not aware of any other cases being reported. There may be other ones, I'm not aware of them. But think of what that might mean. Let me give you a court case. In 1991, Stephen Moberly clearly was guilty of murder, cold-blooded murder of a pizza parlor manager, admitted to the crime. His lawyers, though, when he was being sentenced, requested that he be tested for the presence of that mutated form of monoamine oxidase. Now, was it really his fault, or was it in his genes? What would you have done if you were the judge at that trial? The judge then actually said the law wasn't ready to accept such evidence and therefore rejected the possibility of testing him. And eventually, Moberly went to the electric chair. There are other studies done on the same gene. In a particularly good study in Dunedin, New Zealand. Not on that mutant form, but on ones with low activity. A different form where there was a point mutation that means that there's a slightly lower activity of it. And are they aggressive or not? And this, I think, is the only graph I'll show you, with antisocial behavior up here, and whether they have the low version of that gene or the high version of it, correlated against whether they live in an abusive environment or not. And if you live in a perfectly normal environment, the presence or absence of that particular kind of gene makes no difference. It's only when the gene is imposed on an environment of high abuse does then that it be a better predictor of aggressive and violent behavior. Genes and environment interact together. That dichotomy we're so used to, is it nurture or is it nature? It's both together. Good evidence that those two come together. There are other cases as well where the judge has been asked to relax a sentence on the basis of genetic evidence. The Walter Perez case in Italy was a good example of that. At appeal, he presented data that said he had various mutations in his genome. What would you have done if you were the judge in that case? Would you give a tougher sentence? Because, well, actually, they're always going to be criminal anyway, so you might as well lock them up for longer because they're always going to be dangerous to society. You're going to give a lighter sentence because, well, it's not their fault, is it? It's in their genes. And in that case, the judge docked a few years off of the sentence, saying he was persuaded by the genetic evidence. You could, on the basis of your genome, then be seen to be guilty before you've ever committed a crime. Lock you up because you've got a mutant form of the disease. There was an outcry, it has to be said, in the scientific literature against this. People writing to Nature saying that this rather suggests a form of determinism that we're just not happy with at all. There are other genes that have been implicated in violent behavior. Here's one published in Molecular Psychiatry. I won't go into the details of it. It has, while it may have some element of truth in it, it can be overhyped. So suggesting that we really have no choice at all. And you need to be very careful about this. Here is, and I put the word beware across before I remove it for you. Scientists find that Nazis were correct about heredity and criminality. Taking that literature and misapplying it and suggesting that the new findings by scientists provide proof of what the National Socialist government knew in 1933, that some people should be eliminated. So be very, very careful about how genetic information is used. These associations may work for groups, but they don't necessarily work for individuals. And people who carry that, any form of mutation, can therefore be socially stigmatized. Genes don't turn their owners into killers. There are many people who have that sort of genetic baggage. In fact, that particular gene is present in 30% of Caucasians. So there's not a deterministic link between possession of the gene and violent behavior. As you see, I work for the Faraday Institute for Science and Religion. Maybe I've got a particular variant of a gene that says I'm susceptible to those sorts of spiritual influences, and I'm a mutant. There was a rather bad book published in 2002 called The God Gene, which suggested that a particular form of neurotransmitter storage vesicle, the VMAT2 protein, had a particular variant in those who were religiously inclined. Again, think very carefully about what that means. Here's Carl Zimmer writing in the Scientific American, rather debunking that whole study, saying this is a gene that accounts for less than 1% of the variants found in scores on psychological questionnaires designed to measure a factor called self-transcendence, which can signify everything from belonging to the Green Party to believing in extrasensory perception in one unpublished, unreplicated study. And it's never been put in the peer-reviewed scientific literature. But you can, if you want to have fun, go out there and Google search FunVax. Just have a fun in an idle moment this evening. If, of course, there is a gene that makes a protein that codes for religiosity, then maybe that accounts for some of the extremism we see in the world. And maybe we should vaccinate against it. If you Google search FunVax, you will find what appears to be a CIA briefing of they were going to design a virus that they would use for infecting the Middle East that would inoculate people against religious extremism. It's a total hoax. No evidence for it at all. You see, genetic information does not dictate everything about us. We're not slaves of that information. We have to see beyond a first reaction. We are just the consequence of our genes. This shallow genetic determinism is unwise and untrue, says Walter Gilbert, who was one of the other people who devised methods for sequencing genomes. It takes much more than genes to make us human. I came across this BBC article a year or so ago, where they were testing for the resilience gene. And I have no idea actually what gene that was, looking at a presence of a particular kind of a gene. And they tested Simon Weston. You may remember that survivor of the Falklands crisis, terrible injuries. And they had a long, drawn-out argument. And then the presenter then says to him, so this peak actually suggests, Simon, that you have the gene variant that is not associated with resilience. And yet he's a remarkably resilient person. What do you feel about this? And I think Simon Weston's words there are quite profound. I'm leaning towards being slightly happy about, in the sense that it's a sort of nurture element rather than nature element. I always want it to be who I am, not what I am. It's not what happens, it's what you're prepared to do about it that counts. And even Richard Dawkins would be saying, we alone on earth can rebel against the tyranny of the selfish replicators. So we rebel against our selfish genes, and we have hospitals and social care and all the rest of these that look after the weak and the vulnerable in society. And maybe is genome editing one portion of that, of rebelling against those selfish replicators? Or is it something where we're playing where we ought not to go? Just over a month ago, the Nuffield Council on Bioethics published the first part of its review on genome editing, an ethical review. They make no recommendations in that. That's to come in the second part that will appear, I think, sometime early in 2017. And there are questions you'd ask, well, what about risk? The whole process of modifying has a risk of modifying unintended consequences. What if you modify a gene as well as the one you wanted to? It raises a question about what is normal. There's only one normal person in this room, I'm sorry. That's me. The rest of you are freaks, right? And you should all be clones of me. What a sad world that would be. It could give some form of genetic classism, where those with certain kinds of genetic makeup are seen to be better, superior, more worthy of things. And it could reduce people just to being commodities. Life is just another thing you can purchase off the shelf rather than being real individuals. So here was a number of quotations. There has emerged a global consensus that such gene modifications should be forbidden owing to safety concerns, unprecedented informed consent, in other words, you do it to an embryo who can't make a decision, challenges to human dignity, and the potential for permanent negative impact on future generations, including its abuse for eugenics or enhancement. I'll say a little bit about that in a moment. The parental pursuit of specific traits for non-medical reasons. And the research councils have made various declarations about their response to human genome editing and saying that no, we should not go there for the moment. It has to be, at least in terms of modifying humans, we should stop and think and debate before we ever do this. Genome editing in human embryos using current technologies could have unpredictable effects on future generations. This makes it dangerous and ethically unacceptable. It's interesting, one of the reasons they give for that, though, is we're concerned that a public outcry about such an ethical breach could hinder a promising area of therapeutic development, namely making genetic changes that cannot be inherited. So they're afraid of the consequences of taking science one place where it shouldn't go might have consequences on other places that are funded in terms of research, a kickback, if you like. If we look about whereabouts in the globe could the human genome be modified, and this is taken from an article in Nature, those in the, I don't know, the maroon colour are where it is banned. In this country, it is not allowed to modify human embryos except with specific consent, and then only embryos that are surplus to in vitro fertilization. Other places it's ambiguous, and in a lot of places there's just no law at all. So if you're an ambitious scientist who wants to make your name, you know where to go, because this is cheap to apply. There are all sorts of points for debate on that. This is a list again taken from Nature. Should it be allowed on germlines? Should it just be done on those embryos that are left over from in vitro fertilization, or should you be able to create embryos specially for that purpose? Is the only thing risk involved in this? What if it goes wrong? What about off-target effects, as they're called? Modify the wrong gene by accident. Well, somebody has actually pointed out that the process of making people is a fairly risky one anyway, especially when done by natural means. The number of spontaneous mutations that occur in each generation is probably far greater than would occur by the mistargeting of one of these agents as well. So look at the other side of the coin. Life is risky anyway. Is human genome modification necessary? And in many instances the answer is no. It's an in vitro technique, and there are plenty of other in vitro screening techniques on pre-implantation embryo screening that can detect the presence or absence of various debilitating diseases. There are one or two cases which excuse me, could only be sorted by human genome modification. But here's Francis Collins, remind you, director of the NIH. Advances in technology have given us an elegant new way of carrying out genome editing, but the strong arguments against engaging in this activity remain. These include the serious and unquantifiable safety issues, ethical issues presented by altering the germline in a way that affects the next generation without their consent, and a lack of compelling medical applications. It doesn't get us anywhere that we can't get by other techniques. But what about human dignity in all this? Do we have worth and dignity regardless of what we can and we cannot do? Do we have value as humans because of what we are rather than because of our genetic makeup? C.S. Lewis, writing well before the genome era, talked about man's power over nature turns out to be a power exercised by some men over other men. I think we could put women in there as well. With nature as its instrument, this becomes a means of controlling other people. Could be a means of techno-selfishness, where we modify genomes to make people faster, brighter, stronger. But does that mean better? Probably not. And it's a kind of, this will only be ever used for the rich and the powerful. That means we have the vanity of the rich and the genetic underclass that would come from that. So some simple questions, even if we said it could be done. What genomes in the embryo should we modify? The genome of an embryo that is totally non-viable. That's giving life where there never would have been life in the first place. The genome of an embryo that will have a disease that will manifest itself as soon as the child is born. Something like cystic fibrosis, point mutation. The genome of an embryo that will develop a disease later on in life, whether it's the BRCA2 gene that will give a likelihood of breast cancer, whether it's Huntington's disease that will almost inevitably give that disease as the person goes into their 40s. Is it permissible to modify the genome of an embryo so as to do something cosmetic, change their eye colour or enhance their intelligence? Maybe it shouldn't be written off as playing God, as some people say, because actually we play God every time we intervene by any medical means. This is just another medical means, so they say. But we do need to think very carefully about those people who have some form of genetic disability. Here's Tom Shakespeare, who's frequently in the media. He suffers from a disease, a genetic condition that is effectively dwarfism. And he says that people with disabilities are unlikely to be queuing up for this genetic modification. What they want is to overcome discrimination and prejudice. The voices of people living with illness and impairment need to be heard, not said that they're mutants and therefore should be, whoops, what have I done? And should be corrected. He has that genetic condition. I have achieved everything, he says, I hope for in life, despite restricted growth, career, children, friendship, love. He wouldn't want to have his own genes to be taller, he says. And the disabled cry out, nothing about us without us. They don't feel that they're inadequate. And some philosophers have suggested that people with genetic mutations actually have a moral imperative to transmit those to the next generation, rather than fiddling with the gene pool in humans. I want to dwell a little bit on this girl, Ruthie. She has albinism, which means that she has a very restricted sight. And interviews with her and her parents. Her father saying if he'd had the option to edit the benignness out of her genes before she was born, he and his wife would have jumped at the chance. But as a result of the kind of lifestyle that she lives and the resilience that she's got and the determination, that has now defined her character. So she now goes and plays basketball by virtue of the kind of training she had to go through in order to be able to do that. Changing her disability, he suspects, would have made us and her different in the way that we would have regretted, he says. And her father asked whether wished that her parents had corrected the gene responsible for her blindness before she was born. She didn't hesitate before answering no. Would she ever consider editing the genes of future generations to help them see? Again, she answered without a blink, no. What about deaf parents who want a deaf child? There was a case in the media not that long ago where they wanted to select an embryo that would be deaf like them. They see that as a normal form of personality, a different culture. You might like to think on that one. Is it the slippery slope to genetic enhancement, altered physical ability, changes in musical ability, high IQ? All these multiple genes contribute to them. But is it the beginning of the slippery slope? Can we enhance humans? And if we did it by the CRISPR technique, the one thing it does not do, it doesn't leave a signature of what you've done. Old-fashioned times of genetic modification left marker genes and all the rest of those that said you'd snip something in. This is silent. You wouldn't know that it had been done. What about creating the perfect football team? We know that there are some mutations in one of the collagen genes that makes you predisposed to problems with your Achilles. You could therefore test a child and say whether they're likely to make a good football player or not. You could test your team. Sometimes the parents can be over pushy. Tiger mums. Here's one of my research assistants in Cambridge found this, a case in Austria of the children's race for 3 and 4-year-olds. And you can see what's happening in this race. Now, it's not the kids who are having fun, it's the parents who are so determined that my child is going to win. And the actual children's race is not to be held in 2017. It's the fault of the parents, pushy parents who would want other genetic complements for their children. Enhanced IQ, maybe, if we could understand. There are thousands of genes, probably, no, that's an exaggeration, there are a lot of genes that 74 at least been identified that have some effect on intelligence, even at just a small amount. What is normal? I've already raised that. Human diversity is part of what it takes to make society. And many people acknowledge that the free, unmerited nature of life itself as a gift. We talk about musicians being gifted and athletes being gifted, not something that's imposed on them by genetic modification. Imagine explaining to your teenager that they were modified as an embryo. Those of you who know anything about teenagers, you can imagine what the response might be. You modified me? What? I'm a commodity. I'm not a science experiment. Why can't I be like all the other ungenetically modified girls, as the cartoon shows? And to take just in closing, what if we could actually construct an artificial human genome? Artificial bacterial genomes have been constructed. We've got several orders of magnitude greater, but George Church in the US has suggested that maybe we could make a synthetic human genome, life version 2.0, a writing of the human genome rather than modifying. And technically impossible at the moment. We could, if we did it, we could resurrect the Neanderthals, reconstruct a Neanderthal genome, and then what would we do with it? Would they be museum pieces? It's in the zoo. Would they integrate into society? We wouldn't have the foggiest idea what to do with them, would we? Prediction. My grandchildren will be embryos screened, germ edited, won't change what it means to be human. It'll be like vaccination, says one person from Harvard. Here's another one who actually suffers from cystic fibrosis herself. There are some great things that come from having a genetic illness, she says. Wouldn't modify genomes. So a couple of slides just in closing. The Christmas related. You know who's been naughty and who's been nice. You're not using DNA evidence, are you? Says the child to Father Christmas. Investigating your genome to see whether you behaved yourself rather or were likely to behave very well. And this is what I'm going to wish you in closing. Those of you who know anything about the genetic code and the single letters will know that's histidine, alanine, proline, proline, tyrosine. And in the single letter codes, and so on we go, that is Happy Christmas. Thank you for listening. Happy Christmas.
Questions or observations that have been stimulated by those many thoughts that Keith has planted in our minds this evening? I think you might have stunned them all, actually.
I went on rather longer than I intended, but not too many were asleep.
One over here.
What are your personal views on it, or is that a bit rude?
My personal views is that I think we need to qualify what sort of genetic diseases should we be able to treat. So something like, if you like, a cystic fibrosis, a Huntington's disease, I think their outcomes are clear. And if you like, that's a form of healing, it's a form of medicine, if you like. And so long as we can, in those cases, be sure that we overcome the risk, there'll never be risk-free, but at least the risk is reasonable. In those cases, I think there is a good case for being able to do that. It's a form of medical intervention at a very early stage. But I think we need to legislate quite clearly as to what kind of diseases that applies to. It is going prematurely grey, which I did. Is that a disability? I mean, it's in my genes. I blame my mother and I blame her father. Is that a disability? So at what stage is there the line between cosmetics, really, and enhancement, and, if you like, healing? And restoring somebody to a normal state, and that's where we need to be clear what we mean by normal. I don't see a problem with restoring, if you like, a normal, healthy person. But and that's where the legislators maybe need to go through very precisely to say exactly what conditions are and are not permissible by that sort of technology. So in principle, I don't have a problem if it's to do with diseases where the outcome is serious and it's clear. If it's a predisposition to maybe, possibly, with a 1% chance, then no, it's not worth doing it.
Richard.
I really enjoy that. Thank you very much, Keith.
Thank you, Richard.
One thing, if you want, are we tempering environment and epigenetics within this whole story? And the other thing is that the legislators, and I don't mean that if there are any legislators in the room, there's quite a need for an informed backdrop to allow a true discussion before a decision can be made. And how are we going to be able to engender that debate? Because I don't think that happens at all.
Absolutely, and it needs an informed debate that involves, I think, the scientists who understand the basis of the technology, if you like, the medics who understand the basis of the disease, the ethicists and the philosophers who understand other things as well. And even the science is so complicated. You mentioned the epigenetic effects, and we don't, that's an emerging field we don't properly understand. And this is going to have epigenetic consequences as well, in the way in which our genes are influenced by the environment that we're in. And so there has been a cry, and Jennifer Doudna, who was one of the discoverers of the technique, suggested that there should be a moratorium on that. And I think for the moment I'd support that. It is, at least, that's not a moratorium in doing experiments and finding things out, but a moratorium on applying that to practical human genetics. I don't think we should do it until we're much clearer about some of the things that make us up, what those epigenetic influences are.
Thank you.
Yeah.
Don't you think other countries will take the lead and avoid this debate? And there is a big risk that somebody is going to go ahead.
Oh yeah, absolutely. I'm sure it will happen. There will be a rogue state or rogue scientist who will do it, but we need to have a few thoughts about that. Firstly, is that what do we do with the genetically enhanced person? And my view is they're still a person. So we shouldn't go the other way and say, oh gosh, they're a freak. We don't treat them as a person. But the other thought is, because somebody else might do it is not an excuse for why we should do it first. And there always will be scientific egos. I see, as editor of a journal, incidents of scientific egos that go ahead of the data and actually massage and even fraudulently present data in some places. And the scientific ego is very great. And the first scientist, the first person to genetically modify and bring through to term a genetically modified human being is not going to be forgotten in a hurry. They might not win prizes for it, but as I say, better to be known for murder than not to be known at all. Their name will be there in lights. But that is not any reason as to why we should relax our legislation. We should be pushing for global legislation, enforceable. That's the difficult thing. To agree at what is right and what is wrong, and then to stick to it. But I think in fact, I'm a pessimist. I think it will happen, and I hope, therefore, the outcomes aren't too bad from that.
Another question over
So
here.
potentially sort of leaping a few stages ahead of that, given the scandal in sport this year concerning further sort of drug abuse and all those kind of things that are already quite hard to detect, do you think it will be not too distant future that this is part of the have people genetically enhance themselves as well?
Yes, I mean, the whole gene doping thing is part of that. to look even further down the line, the parents who genetically modify the embryo so as to go for fast or slow muscle types, so as to give a particular form of athleticism. And as I say again, with this technology, it could not be detected. It would just be, there's no marker genes that go with it at all. It just happens to be that collection of genes in that individual. The child grows up, it's all kept quiet, nobody knows, there's no means of detecting it. You can't do a urine test to show that they've been genetically modified. So the ethics of sport are going to come into this. It's still not a deterministic. You can have a child who has all the genes for physical prowess who is still a couch potato, and they're not going to win many medals that way. So there is that element of personal decision within that. But I think there needs to be, in the same way over drugs and sport, there needs to be a big debate over genes and sport. And then, of course, it comes back and say, well, you know, those people who are naturally good sports people are gifted with those particular genes. Is that right that they should have it and I don't? Well, to my view, that's just the wonderful kaleidoscope of life that makes us different people.
You gave a number of examples of where the press in particular had made mistakes and misunderstandings and so on. Is there something that the community who understand the science could or should be doing to put that right, or is it a lost cause because the science is just too complex?
I think the community does have a responsibility for making sure that the media report it carefully. I think there are a number of things at stake here. Is that it's in the scientific ego. If you want to make a news story out of it, and news stories bring grants in eventually, if you want to make a news story out of it, to say a gene has a 0.1% influence on particular trait is not going to make any big press. If you read the small print and you read the Times and the Guardian and maybe the Telegraph, you will find usually those sorts of disclaimers, but it's not in the banner headlines at all. So yes, I think we do have a responsibility for making sure that the press report these things properly. And actually, even amongst scientists, understanding the terms. So we use this word heritability. Now, I'm not going to go into the definition of what that means because I'd probably get it wrong. But the heritability is to do with the percentage variance that's associated with a particular gene, not the penetrance of the gene itself. So, and it's not quite the same word as the man and woman on the street understand by that, and the media can pick that up.
In plant genetics, we have this difference of the genetics times the environmental impact, giving quite different characteristics as a result of that. And therefore, you have a very random element which is out of the control of the human being. Are we going to find in human genetics that this E factor may always, in the end, just beat the scientist from being able to do his prediction as he expects?
I think that's a very good point, and we don't know. Yeah, the environmental impact we can begin to assess with plants and other organisms. You can do test experiments. You can't do that with humans. What would be the environmental impact? We don't know. I mean, on a very different level, I was reading, was it today, yesterday's paper, that claiming that humans were evolving in terms of the effect of caesarean sections. Now, that just seems too rapid to me, but it was there in the press anyway. Changing, if you like, our own genetic makeup, our own environment, the way in which we, in childbirth.
Okay. Keith, thank you very much indeed for a very stimulating evening. I think you threw so many different things at us, so many different ethical dilemmas and so on. There's plenty for us to be thinking about over as we go home and so on. But I think we're extremely grateful for the pearls of wisdom that you have shared with us tonight. And we've got a small token just to say thank you very much. Let's just thank Keith yet again.
Thank you very much. Thank you.